it's really my pleasure to introduce Professor Adrian Hill. Professor Hill is really a champion of molecular medicine for healthcare and health improvement, especially in the poorer regions of the world. He is the director of the Jenner Institute at the University of Oxford. The Jenner Institute, as many of you may be aware, focuses on developing vaccines for infectious diseases, especially those that are of high prevalence in the low and middle income countries. Professor Hill is a world-renowned vaccinologist and is the Lakshmi, Mittal and family professor of vaccinology. His own long and sustained research on HLA polymorphisms and susceptibility to malaria has resulted in the most promising vaccine for malaria, which is undergoing clinical trial in Africa. In addition, he's deeply engaged in developing vaccines for tuberculosis, sepsis, rheumatic heart disease, and so on. In 2014, Dr. Hill's group led the first clinical trial of an Ebola vaccine. I like to remember Professor Hill as a human geneticist. He initiated his career in human genetics. Still, he is intimately connected with the human genetics group at the Wellcome Trust. And his work on thalassemias is something that we all remember very well. So overall, Dr. Hill is a human geneticist, but has actually specialized in vaccinology. He is very well decorated. Last year, he was elected to the Fellowship of the Royal Society. He was appointed an honorary Knight Commander of the Order of the British Empire. So I'm not going to take too much more time and like narrating all his accomplishments, only some of which I know. It's been a pleasure for me to know Professor Adrian Hill, again through human genetics predominantly, but also through a little bit through vaccinology. He today has kindly agreed to deliver a Frontiers of Science lecture. And the title of his lecture is Development of a Simian Adenovirus Vector as a COVID-19 Vaccine, Progress and Perspectives. The Jenner Institute, as all of us know, and Professor Hill in particular, has been very involved in this particular Oxford AstraZeneca vaccine that's known as Covishield in India. Without further ado, let me invite Professor Hill to deliver his talk. Adrian.
Thank you very much, Professor Mujumdar, for that kind invitation, and particularly for inviting me to give this talk. It's been a pleasure giving talks on COVID-19 vaccines over the last year or so, because the audience is so well informed on the background to this subject. So I'll try and not cover just things that you already know, but give you a perspective of the director of an institute that was really trying to move very quickly to develop a vaccine against this disease, and particularly to develop a global vaccine, which has had its own challenges, some of which I will share with you. So the title is Development of a Simian. That's from an ape species, actually a chimpanzee, adenovirus vector as a vaccine against COVID-19. And I'll tell you how we got to where we are and have some reflections or perspectives towards the end. As you heard, I'm at the Jenner Institute in Oxford, which is one of many institutes in the medical school. I founded that in 2005 to bring together quite a few groups that were already very active in vaccines in the university to try and progress more quickly. And this was a great opportunity to show what we could do with everybody working together on a single vaccine. This slide from a couple of months back summarized the position that you know well. Hundreds of millions of clinical cases of COVID-19 caused by SARS-CoV-2 had already been recorded by then. We're now, of course, over 4 million deaths that have been counted. One suspects that there are many more than that have not been in that total. A huge number of vaccine candidates, not just designed and being tested in animal studies, but in clinical development. And this will, I believe, for decades be the great example of comparative vaccinology, looking at the same antigen the same vaccine being targeted by a great variety of different technologies that will be useful for other fields that many of us care greatly about, like HIV, malaria, TB, and other areas needing new vaccines. 10 of these new vaccines for COVID-19 are now licensed and being used in at least a few countries, and there are some others licensed just in one. We'll come to that at the end. But of course, the bottom line here is this has all happened at totally unprecedented speed. If I had written a grant application saying we'd like to make a COVID vaccine in a year, 2 years ago, it would have been turned down as totally unrealistic and just unprecedented. And of course, the reviewers on those points would have been correct. So the problem we were faced with back in January of last year, when we first heard about this Wuhan virus, we're aware that it was a coronavirus. And of course, because the Jenner Institute is interested in veterinary vaccines as well as human vaccines, we knew well that coronaviruses can cause great problems in animal species. Vaccines had been developed in animals against coronaviruses. We also had already been working on MERS. Sarah Gilbert had made a candidate vaccine for Middle East respiratory syndrome, a coronavirus relative of SARS-CoV-2. We were aware that in 2003, we were lucky that the first SARS virus only went to a small number of countries and could be controlled relatively quickly by traditional means. But this looked as if it might be different, and very soon it was apparent by the third week of January that there were many asymptomatic cases who were infectious from work in China, and that a vaccine was going to be needed. So basically, simplifying, there were 4 vaccine technology platforms that you might have adopted. And what I'm trying to do here is give you a sense of some of the known advantages and disadvantages of the different vaccine types, and we had experience with all of these in the Jenner Institute, and why we plumped for the adenoviral vector technology that I'll spend the rest of the talk on. Now, if you'd ask most vaccinologists what is the standard vaccine technology, they would explain to you it's expressing a protein, a subunit vaccine, adding in an adjuvant and immunizing several times to get good antibodies and protect against a disease, which is true, but you don't get any CD8 T cells. We suspected they might be important against a viral infection of the upper and then lower respiratory tract. You have to be able to express that protein in the correct conformation. That's not critical, as we've seen with some of the world's largest vaccine companies, GSK and Sanofi, working together to try to do that quickly. It hasn't been easy. Of course, you need an adjuvant, too little time to develop a new one, and you will need multiple doses. This will not work as a single dose, is the experience. Particularly in China, but also in India and other countries, there have been very impressive efforts to grow the virus itself, inactivate it, as Louis Pasteur might have done over 100 years ago, and vaccinate with that inactivated virus. The risk there, of course, as famously with Zika, is that the virus may not grow very well. And you don't know how well it will grow for many weeks or even months, certainly in a large bioreactor, and your yield might be very modest. Turns out that the yield has not been too bad of SARS-CoV-2, and companies like Sinopharm have made huge numbers of doses. So that approach has worked reasonably well, but the efficacy may not be as high as with the other vaccine technologies. The more modern vaccine types, RNA, of course, famously very new, and DNA, which has been around for over 20 years. DNA has a problem with strong enough immune responses to be protective. RNA was little known at the beginning of last year. It had been around for 20 years in the research stage, but there was no proof of concept that any RNA vaccine worked in humans. We already knew about thermostability challenges, a big issue for many countries in the world. It wasn't clear whether you'd get away with one dose. You can actually now, but that is news. Multiple doses were considered the standard. Manufacturability really was a question mark, and the cost excuse me, is relatively high, and certainly was unclear in January of last year. And therefore, there was a bit of a risk with RNA, and certainly the Jenner Institute was not in a position to manufacture. So what we plugged for was a technology that we had in-house, had been working on since about 2004, had in the clinic since 2007, initially for malaria vaccine development. And I'll take you through some of the features of this. It's using another virus as a viral vector, an adenovirus. Initially, way back in the 1990s, adenovirus 5, a human adenovirus, was known as a vaccine technology, but it was soon identified that getting antibodies to adenovirus 5 from natural infection might interfere with the potency of this approach. So one of the highlighted points here is that a simian adenovirus avoids that issue because we're not exposed naturally to simian adenoviruses. It's a non-replicating vector. We make it non-replicating by taking out some key genes in the E1 and E3 region. That's all straightforward, had been very much done before, but takes a little bit of time. You then go and put your transgene, if you want to call it that, for the antigen you want to vaccinate against, in this case, the famous spike protein of the coronavirus, into the genome, into the 35 kilobases of the adenoviral DNA, with a very strong CMV promoter. So that's the predominant antigen expressed by this virus. And what you get are strong B cell and particularly T cell responses after a single vaccination. The T cells are relevant here because that's why we had been developing adenoviral vectors, because you can protect in principle against HIV and malaria and TB by T cell induction. But of course, there are very few licensed subunit T cell vaccines against any disease. We were not alone in doing this. We did the early trials in 2007. By the start of last year, 16 different chimpanzee adenoviruses, I'm going to call them CHADs from now on, had been in clinical development, not just by the university, but by big companies like GSK and Pfizer. We brought in AstraZeneca, I'll come to that, and we co-founded a spin-off company from the university, Vaccitech. So about 6,000 people had been immunized by various chimpanzee adenoviruses before 2020. Interestingly, the majority of those in Africa, because the programs being prioritized were malaria and Ebola. More broadly, other adenoviral vectors had been given to well over 100,000 individuals since the first trial in 1991. So we felt reasonably confident that this would be a safe technology in fairly large numbers. And as I've already said, you get very good CD8 T cell responses, as I'll show you in a moment. This is just some background on what we already knew. This was work on Middle East Respiratory Syndrome coronavirus from Sarah's group in a clinical trial that I was the PI of in Oxford. And you can see here that with different doses, you do get neutralizing antibodies. With the higher, now the standard dose of the vaccine, you get those in most individuals, as you can see here. And we already had a time course. So if you look at the bottom right-hand panel here, here are the immunoglobulin G antibodies at those different doses, not hugely different, particularly after a year. So we knew that you got sustained antibodies that had neutralizing capacity with a single dose. And of course, in an outbreak situation or in a pandemic, I would argue that single vaccines should be preferred. We also, of course, got T cells. This is what this technology was developed for. If you look on the y-axis here, you're counting the number of T cells specific to the spike protein, actually for a mixture of peptides, induced by a single-dose vaccine in the same trial. These peak at about 14 days with lots of activated effector T cells, both CD4s and CD8s, similar proportions, and again, reasonably well sustained at a year. So that encouraged us to adopt this platform for SARS-CoV-2. Back at the beginning of last year, and you don't hear much about this now, there were huge discussions about the safety of vaccinating against SARS-CoV-2, convened by WHO. There had been evidence that with the first SARS virus, you could see immunopathology if you vaccinated certain animals in certain ways. and this looked like antibody-dependent enhancement of infection, similar to what you sometimes see with dengue. Now, in the event, this has not materialized, it's hardly discussed these days, but that was important for our development plan in that we felt we had to vaccinate at least some animals with this vaccine and infect them before we started dosing humans, because of course it would have been a total disaster if enhancement had been seen with these new vaccines being developed so quickly. So we were lucky to be able to do that with an excellent group of collaborators at the NIH facility in Rocky Mountain Labs in the United States. And what you're looking at here is a brief summary of a published paper in non-human primates, where some of the animals have been vaccinated. Those are shown in red. The control groups vaccinated with GFP in blue do not have antibody titers. The vaccinated animals do have neutralizing antibodies and T-cells. But importantly, we were able to rapidly challenge those animals intratracheally, intranasally, orally, and ocularly. Perhaps slight overkill, but we wanted to be sure the vaccine was safe, and observed that there was no evidence of increased disease in those animals that had been vaccinated compared to those who had not. So that set the stage for starting the first clinical trial in Oxford. Of course, what I've left out a moment, and we'll come back to later, is the need to manufacture the vaccine, which we were able to do very quickly because we have our own manufacturing facility in the university, which we have access to and control over, and we're able to generate a vaccine in about 4 to 6 weeks at that facility, ready for the 23rd of April last year, which was the day after we had seen the safety data from the non-human primate studies, vaccinated our first volunteer, surrounded by cameras and global media interest, and it all went fine for about 2 or 3 days until I was called by my collaborators in Pune on the 26th of April saying, what is this report? It's all over social media, saying that this poor lady had died. Colleagues were, or other vaccinees were in intensive care, and it was all due to the vaccine. So of course, this was completely fake news, but it was a stark introduction to what we've got used to over the last 18 months and never had to deal with before, which was the intensity of the media coverage of this whole program, with people being very strongly supportive or very much opposed to what we were doing, anti-vaxxers and so on. And we've just had to live with that. And I'll tell you a little bit more about it as we go through. But by the 26th of April, happily, this volunteer, being very media adept, just filmed herself with the morning newspapers and pointed out that she was very much alive, and that seemed to help with that particular incident. But what were our chances of making a vaccine last year and having it licensed, which of course happened by the end of the year? The great experts in vaccinology were not totally, well, they were pretty unanimous, but they were unanimous in thinking it could not be done. And they had a lot of evidence on their side. Tony Fauci in the United States saying, in April, I reckon 12 to 18 months minimum for a vaccine at the earliest, he said. His colleague Paul Offit, who writes the huge textbook on vaccines, told CNN when Tony Fauci said that, 12 to 18 months, I thought he was being ridiculously optimistic, and I'm sure he did too. So that wasn't encouraging. But of course, one of the things we were trying to do by attracting publicity as an academic group was to interest industry. And normally it takes us years excuse me, to attract an industry partner, if it can happen at all, with one of our academic vaccines. We wanted to see if that would help. And of course it did, because on the last slide here, by the 30th of April, we had an exciting partnership with AstraZeneca, one of the world's leading big pharma companies selling flu vaccines in the UK and elsewhere. And they, of course, have been critical to the progress of what's been called the Oxford AstraZeneca vaccine. I'll come back to later that we already had an Indian partner who turned out to be, I would argue, at least as important as AstraZeneca in this whole development program. And by the end of April, they were already making some of the vaccine in Pune. But in the first clinical trial, briefly, we saw that the vaccine looked relatively safe. very similar to all other vaccines. Some sore arms and fevers, but overall satisfactorily tolerated. Neutralizing antibodies were induced with a priming dose. You can see that here on the y-axis, day 56, that level of neutralizing antibodies. But importantly, if we give a second dose, which we explored, you get 3 times as high antibody titers. And this caused a change in our plans because we had no idea, and nobody else had either, what level of antibodies you would need to protect against this virus and disease in humans. But we thought 3-fold higher is likely to be better. So we moved from a single-dose vaccine program, which Johnson & Johnson have persisted with, and of course they're making an adenoviral vector vaccine and using it as well. But to increase our chances of success, we moved to largely a 2-dose program. We compared those titers to individuals who'd had COVID and had recovered, convalescent plasma samples, and they happily were very similar levels. So we thought this vaccine might work. If you look at the T-cell responses, a feature of this vaccine technology, very good T-cells by ELISpot assays at day 14. And you can show by flow cytometry that about half of these are CD8 T-cell responses. And for the immunologists, secrete a whole variety of cytokines, particularly Th1 cytokines, which is what we were looking for. So jumping forward quickly, we moved from a phase 1 trial starting in late April to a phase 2/3 trial starting in late May, just a month later, which is extraordinarily rapid. And we were probably fortunate in that our regulator in the UK, the MHRA, had seen a lot of adenoviral vectors, particularly chimpanzees, that we had taken into the clinic, about 10 in total for other indications. So they were relatively happy that these vectors were safe, even though they were relatively new. The UK trial for phase 3 was designed to enroll about 12,000 subjects across 19 sites in the UK. So we had a fantastic collaborating team in the UK alone, but more collaborators in Brazil, 11,000 subjects, 6 sites across that huge country, a smaller number in South Africa. In the US, AstraZeneca led on a 32,000 subject phase 3 trial from the end of July. And in India, there was a bridging study led by the Serum Institute of India in 1,600 subjects. So by the end of the year, we had over 60,000 subjects dosed with a very satisfactory safety profile. Now, there's been a lot of discussion and some negative comment about the design of these trials. They were rapidly put together. They were coordinated, of course. We wrote the protocols for all of those. But what we did include were, and some people commented on, was having a lot of different dose levels, which we wanted to do. You want to know, do you need a high dose of vaccine? If half a dose would do, that would give you twice as many doses for the world. Did you need 1 dose, 2 doses, 3 doses? Do you need a booster? We're still working on that. What should the interval be between the priming and boosting dose? That turned out to be really important. I'll come on to that in a moment. Would you see efficacy after a single dose or after only 2 doses? What were the immune responses like? And what we did almost uniquely is we tried to measure efficacy against asymptomatic infection. So we got our amazing volunteers in the UK, most of the 12,000 of them, to swab their throats every week for up to a year. We accumulated about 250,000 swabs doing that, and were able to get a readout on whether the vaccine protects against asymptomatic infection. But of course, we now know it does, but not as well as against disease or against severe disease. But despite this complexity, which we built into the program, regulators want a simple primary endpoint. And we did identify that for them, discuss it with them, and agree with different regulators what that endpoint should be. And happily, by the end of the year, we had over 500 subjects to analyze who had developed infection or disease. And that was a very powerful study. So what do you actually measure? Well, most importantly, you measure prevention of severe disease, hospitalization, and death. The numbers were straightforward, 16 in the controls, none in the vaccinees. The most commonly measured, and the one that makes the headlines, is protection against mild clinical COVID. Symptomatic disease, these are nearly all mild to moderate cases. I'll come to that in a moment. And I've already mentioned efficacy against asymptomatic colonization of the upper respiratory tract. That was about 60% in one of the major groups. So the headline result ended up being 70%, but that was a little bit misleading because it was a compound of two different immunization regimes, one giving 90% efficacy and one giving 62% efficacy. Now that's not a bad thing. Of course, you're allowed to pick the regimen that is better. So this was one where most of the individuals had a low dose, a half dose followed by a standard dose. But more importantly, they had a 12-week interval between the two doses. And that turned out on further analysis to be the only driver really of higher immunogenicity and efficacy, having 3 months between the priming dose and the boosting dose. And just to put this in perspective, what regulators were hoping for most of last year was a 50% or more efficacy for licensure. So we easily hit that level. Rather gratifyingly, but also slightly frustratingly, a month after we had done all that work and reported the efficacy data in November and December of last year and got regulatory approval by the end of December, a beautiful study of data linkage, first in Scotland and then later in England, simply followed up 4.5 million people. 500,000 of them given the Pfizer vaccine that had gone forward in parallel, or our vaccine, and therefore 3.5 million with no vaccine. And by linking the records of who was hospitalized with who had received particular vaccines, you get a readout very quickly and inexpensively on the efficacy of the vaccines with a single dose against hospitalization in that population. And I think this was really important because it showed single dose efficacy. It showed efficacy really in large numbers for the first time in mainly older adults who were prioritized for vaccination at the end of last year. And similar data has appeared from other studies in England. Nice comparative studies now, one in Bahrain just the other day, comparing the Pfizer and AstraZeneca vaccine. You're seeing very similar overall efficacy, particularly with a single dose. You may get better efficacy after 2 doses with the Pfizer vaccine, depending on the context. So approvals came thick and fast. The UK first by New Year's Eve, essentially last year. For those over 18, that's now been extended 4 to 12 weeks apart. 12 weeks recommended based on the data I've just shown. And a single dose being useful. If it weren't effective at 76%, you might have concerns about waiting 12 weeks for the booster. So it was good to see good efficacy with single dose. And then 82% efficacy overall in the UK with the 2-dose regimen. Just summarizing what has happened since, of course, there's been a large US trial, 32,000 subjects. Again, almost complete efficacy against severe disease and hospitalization. Very good efficacy in older adults. And this was a 4-week prime boost interval, and still 76% efficacy was seen. And by the last day of last year, there was regulatory approval in 5 countries and more very soon afterwards, crucially by the WHO and the European Medicines Agency. And by March, we had approval in over 100 countries and already been the major supplier of COVAX, that crucial facility convened by the WHO, CEPI, UNICEF, and others, supplying vaccine to many low-income countries. By March, the Serum Institute of India, our largest manufacturer by far, had distributed over 120 million doses, and just in the last few days, I'm told they have now manufactured around about 1 billion doses, which is an extraordinary performance. I'll come on to that. The European Medicines Agency in March noticed this very rare possible safety signal then. It's definitely a safety signal now, cerebral vein thrombosis, but had a very low frequency. And interestingly, for the geneticists amongst you, it appears to be hugely different in incidence between different populations, mainly affecting Northern Europeans of white ancestry. So there are a lot of ongoing trials. A homologous booster, a lot of discussion about who should be boosted and whether older adults should be boosted in the coming months, particularly in Europe. Safety and immunogenicity has been shown in children. We've done a lot of prime boost regimens, putting two different vaccines together, like AZ followed by Pfizer, but all of these vaccines now in a comparative trial in the UK. I'm excited about giving this vaccine intranasally. Of course, adenoviruses infect you intranasally. So would that be a good route of administration? We're looking at that carefully at the moment. Won't happen this year, but might be an important route going forward. We have a variant trial in progress. This has now started of the South African variant. And of course, we're working on the Delta variant, and that will come along too. A brief slide on the safety data. And I know I'm talking to some people who are very numerate and with statistical expertise. And our feeling here is this has been over-egged as a safety concern. There was huge media interest, some of it coming from the financial press in the US, who were not exactly positive about our vaccine all along. There's no overall increase in thrombosis. People need to be aware of that. There's a very specific and very rare syndrome, vaccine-induced thrombotic thrombocytopenia, at about 5 per million in Northern European vaccinees, compared to a background rate of about 1. It's only seen with the first dose, not with later doses. That may be due to anti-vector immunity, knocking it out. It is associated with antibodies to a platelet component, platelet factor 4. And all the regulators have been entirely consistent in saying that risk-benefit ratio is strongly in favor of vaccination. That incidence of an important side effect is seen with other vaccines that have been used for decades. But the policy decisions from the expert groups, many of them academics in different countries, have been hugely different from different countries in Europe, from the EMA, particularly Denmark and Norway and Switzerland, initially banning the vaccine and then restarting. But one needs to keep that in perspective. We are working on the mechanism. I won't spend much time on this, but I think this is addressable. And there is an interaction between the vector and platelets that we can ameliorate in a variety of ways, perhaps as simply as using a lower vaccine dose. Then variants appeared, of course, first the Kent or UK variant, Brazilian variant, South African variant. South African being the most problematic in that antibodies induced by the original Wuhan strain are 9-fold less numerous in neutralizing assays against the South African virus than they are against the Wuhan strain. Brazil, not as bad. UK variant, we have very good efficacy against. That was shown in clinical trials. And then famously, the Delta variant, first identified in India, which is now the globally predominant strain, and one that, again, the vaccine is efficacious against. It's somewhere between the Brazilian strain and the South African strain. So just one slide on that. We first heard about this from our colleagues in Pune. It's become predominant not just in the UK, but in many other countries. It is more transmissible, but not clearly more virulent. We see reduced neutralization, but it's not as marked, as I said, as against the South African variant. And current vaccines, I say, appear to be protective against severe disease. They certainly are, but perhaps not quite as protective as against the original strain. So just to summarize briefly, where a whole variety of other vaccine and vaccine groups working in parallel with us have got to. So remembering those 4 classes, actually the most frequently licensed vaccine has been viral vectors. Adeno5, the human adenovirus vector from CanSino, licensed in China, not very much manufactured or distributed. Our own vaccine I've talked about. Ad26 from Janssen, single dose efficacy of 67%, licensed in the US, but not as widely distributed as the ChAdOx1. And the Russians, of course, came along with an interesting prime boost regime using 2 adenoviral vectors. That's a very good approach technologically. You get better immune responses, both T cells and antibodies, with 2 different viral vectors than with 2 doses of the same, and indeed they get good efficacy, but the manufacturing challenges are of course much greater. We're trying to make 2 vaccines, at least one of them with replication competence. Famously, the Pfizer and Moderna vaccines came along and showed efficacy in November, a couple of weeks before our vaccine in late November, and their efficacy is very impressive, at least against the Wuhan strain, 95% to 94%. These are not really comparable to the efficacy we were seeing of 82%, because a more severe categorization of disease was used by Pfizer and Moderna, we believe, and certainly by Janssen. So be careful just comparing those headline numbers. DNA has had a bit of a struggle. Inovio are not there yet. They're in phase 3. But just this week, Zydus Cadila in India, of course, have had approval from DCG, I understand. And CureVac, the original RNA vaccine company going for over 20 years, had lowish efficacy, 47% in phase 3, and that's not in use. The inactivated virus vaccines have done rather well, particularly to manufacture so much vaccines, Sinovac and Sinopharm. Not clear exactly how comparable the efficacy is of these numbers, but it's certainly over 50%. Bharat have reported 81% efficacy, and of course are licensed in India. And Valneva in France are close to submitting for licensure for their vaccine. And sadly, the original vaccine type, protein and adjuvants, not licensed yet. Novavax have fantastic clinical trial data. They have an excellent adjuvant vaccine, but they haven't had the approvals they need to distribute just yet. And for those of you who have nationalist tendencies, and if you have, I would discourage you, just a reminder that the world really has contributed to this extraordinary effort. There are 10 vaccines widely used and licensed, 4 from China, 1 from India, Russia, Germany, UK, Netherlands, and the USA, all contributing 1/4. So just to finish up, I'll talk about 2 things briefly. Some other activities that we've been undertaking that I think might be quite important in the long term, and then a few slides on manufacturing. We have developed, slightly controversially, a human challenge model in the UK. No other country has done that. It's happening at 2 centres, Imperial College London and Oxford, giving tiny doses of the virus to people who've either been either recovered from the disease or are immunologically naive, and treating them very rapidly. And so far, so good. I understand almost 50 people have now been challenged, and the trials are going forward. That could be very important for rapid licensure of new types of vaccine. And of course, people are still trying to develop better, longer lasting, more effective, more universal coronavirus vaccines. New routes of vaccination I've already mentioned, intranasal. There's also a trial of aerosol starting up soon. There are single trials going on in South Africa. Other companies are looking at oral adenoviruses given as an immunization route. And I think that's fantastic for the field. And I suspect some of these are going to be very useful and easier to deploy than an intramuscular needle injection. And no matter what happens on COVID, the world academically has unprecedented comparative data. It, until 2 years ago, was extraordinarily difficult to get 2 companies to compare their vaccines for the same disease in any meaningful way. There were incentives preventing them doing that. Whereas now, I just showed you a trial of 7 different COVID vaccines being compared head-to-head in the UK from 7 different. Never before had so many technologies been compared for efficacy, speed of development, immune response, magnitude, and quality, and of course now durability. A lot of discussion as to whether the RNA vaccines might have less durability of their immune responses, even though they were initially very high, and that's being looked at very carefully. So what about manufacturing? A really bad starting point is to realize that never in the history of the world had more than 500 million, that's 500 million doses of any vaccine for any disease being distributed globally. And of course, for COVID, you'd quite like to have 2 doses for at least all the adults on the planet. So maybe 10 billion doses if there are 7.8 billion people and you're vaccinating the adults. So we wanted to go suddenly from never 500 million to 10 billion, a 20-fold increase. Remember, with a set of new vaccine technologies, principally adenoviral vectors and RNA, that had never been manufactured at the million-dose scale before. So that's quite an ask when you remember that it typically takes you 2 or 3 years to get a new vaccine technology or even biological technology at a level that you would get licensure for. So that's what people were seeking to do. We knew that demand would be immediate after licensure. It was. And CEOs of various companies said, yes, we'll make 1 or 2 billion doses in 2021. What they didn't make entirely clear was that you wouldn't have a quarter of those 2 billion doses in the first quarter and a quarter in the last quarter. Of course, you ramp up in manufacturing. So there weren't enough doses. Some of our 22 manufacturing facilities had batches that failed. That's what happens, particularly when you go as fast as you can. So what we did in the university way back in February of last year was develop a distributed manufacturing plan. Particularly Sandy Douglas was central to this, one of our leaders on manufacturing technologies. We started manufacturing at 3 UK locations in the first quarter of last year, also in Italy, Netherlands, and brought in a Chinese manufacturer and an Indian one, famously the Serum Institute of India. And then when AstraZeneca came in from May, they added 20 further facilities in 15 countries. So we made best efforts, you might say, to produce as much vaccine as possible. This is an AstraZeneca slide showing the distribution of those facilities in different continents, deliberately so that if one big country decided to not export any vaccine, and that certainly happened, you'd have other manufacturing facilities that could try and pick up the need for other parts of the world. And the goal by AstraZeneca was to make up to 3 billion doses. I'm convinced we'll make over 2 billion doses. I'm not sure we'll quite hit 3 billion this year with all of our partners, but it's still been an extraordinary effort. I like showing this slide. This is what we really set out to do, to make a global vaccine. Today, and for many, many months, the Oxford AstraZeneca vaccine has been the most widely distributed. Today in 184 countries and counting, with 3 notable admissions, of course: the US, where matters have been complex, Russia and China, which has billions of doses of its own vaccine. The next closest is Pfizer at about 120, and then all the others in the tens rather than hundreds, and Sputnik catching up fairly quickly. There were 650 million doses of this vaccine made by the end of May. It's now of the order of 1.3 billion doses by my own count, putting different numbers together from different places. About 1 billion of these made at the Serum Institute of India. It is by far the lowest cost vaccine still, at about sometimes less than $3 per dose, but $3 per dose for low-income countries and for the rest of the world at the moment. And it is the major vaccine for the COVAX program. So to that extent, we succeeded in trying to supply countries where we knew there would be a difficulty in providing adequate supply. We didn't completely get there. If you tell me where you live, I'll tell you which vaccine you're likely to have. That's what this slide shows you. When the first 2.8 billion doses had been delivered, and when you're at 5 billion now, if you were in America, you were going to have an RNA vaccine. If you were in India, you were very likely to have the Oxford AstraZeneca vaccine. If you live in China, very likely to have the Sinovac or Sinopharm vaccine, and so on and so forth. But unfortunately, this slide a couple of months ago, 85% of doses delivered had been in high and upper middle income countries. Only 0.3% of doses had been administered in low income countries. So there's a huge inequity problem. We saw this coming. It was really difficult to do things about it. It'll be difficult again, but we have time for the next pandemic to really ensure you don't have 74% of people vaccinated in North America and less than 4% in Africa 6 months into the vaccination campaign. But just a few reflections to finish with. COVID certainly changed the way we make vaccines, broke all the normal rules. Took 10 months, not 10 years, to license a new vaccine. We had an amazing amount of money. We struggled to do that, to get funding for diseases like malaria. We had academics as well as biotechs, as well as pharma companies making their own candidates. And we had that extraordinary manufacturing effort I've just touched on. What helped, I'm asked? Well, having done lots of vaccine trials before, so it allows you to go faster. Having fantastic experienced trial sites and trial centers that we could call on for our program was crucial. A regulator that was supportive of moving faster than even that very fast regulator had ever moved before. Safety data on adenoviral vectors were crucial to moving that fast, even though they were often in hundreds of people and not in tens of thousands or millions. And doing efficacy trials in many countries meant that in the middle of last year, when there was very little COVID in the UK, we were still getting efficacy data from Brazil. I've emphasized that what was most problematic and remains a problem is manufacturing capacity in the world. It's great to make a good vaccine. If you can't manufacture it for the world, you've got a problem if there's a pandemic. No one knew how fast a vaccine could be developed, even the Lumini in the United States I mentioned. There were hugely different requirements from different regulators. For example, in China, vaccines were licensed without efficacy data in some cases, in Russia as well. Is that okay? I don't know. I think it could be. Many ethical issues unresolved, including that one. A ridiculous amount of nationalism, countries saying, do a trial in my country or we want to prove your vaccine in this pandemic. Well, why? Not clear to me. Okay, so last slide, some conclusions. 10 COVID vaccines have been developed, licensed. 3 were available by the end of last year. The Oxford viral vector vaccine has some advantages. I won't read through those again, but global supply, particularly for low-income countries, has been one of our strengths. Manufacturing scale-up, as anticipated, was a greater challenge than making the vaccine and showing it worked. Press coverage overall was wonderful, but often misleading. There's a separate talk I could give you on that. And we know now that most vaccines for important diseases like TB and malaria could be developed much faster. And hopefully, in the future will be. So I will stop with one inadequate acknowledgement slide, just showing the PIs in Oxford who are various aspects of this development program. Andy leading the clinical trial program, Sandy on manufacturing, Tess and Sarah on, Tess and Katie on immunology, and most important of all, Sarah Gilbert, my colleague for the last 25 years, leading on the making the vaccine and its development. Thank you very much. I'll be happy to take questions.
Thank you very much, Professor Hill. That was just absolutely wonderful lecture. There are a large number of questions already. The first question is, if half dose compared to the full dose gave better efficacy, why wasn't it approved for vaccination?
Yeah, good question. It wasn't the half dose that was causing the better efficacy. It just happened that in the 2 trial groups that we had efficacy data on, all of the, or nearly all the people with the half dose followed by the full dose had a long interval. And when you analyze the data, it's having a long interval between the first and second dose that increased the efficacy, not having a half dose. In fact, a half dose of its own after the first administration is not quite as immunogenic as the full dose. So that was that was a misinterpretation by the press. We never said it was the half dose that was causing that, and within weeks, we and others were able to clarify that it was the long interval, not the half dose, that improves efficacy.
Okay, the next question is, how does HLA polymorphism contribute to the vaccine efficacy?
That's work in progress. We don't know as yet. We know that a variety of genetic factors impact risk of severe disease. Surprisingly, ABO blood group in severe COVID. But because this is a very large antigen with very large numbers of T-cell epitopes in the full-length spike protein, we're not using the receptor binding domain, which is a small fragment, we're using the whole spike protein. We don't expect enormous effects of HLA, but that will take a little while yet to define.
The next question is not directly related to your lecture, but nevertheless, I'll mention the question. This is about the origin of the virus, of the coronavirus. Was it a lab leak? Does it lead to biological warfare? Was it animal to human transmission? That's the kind of, it's a long question, but that's exactly what the person is asking.
Yeah. My own view is it's extraordinarily unlikely that it could have been a deliberate bad behavior by any human group or lab in China. You just wouldn't do it that way if you were trying to make a nasty virus. Look, the reams have been written on this. A summary that's very approachable and very clear I would refer you to is the new book on Spike, called Spike, on the history of the pandemic by Jeremy Farrar, the director of the Wellcome Trust. Look, it's not totally impossible that this was made by some human, but it just seems totally implausible that if you were trying to make a very nasty virus that you would do it this way. It's massively more likely that this is a virus which we know has close relatives in other primate species, and that came from zoonotic transmission in China at the end of 2019. But it's a very long answer, and lots of expert groups have looked at this. But my own view is that it's very, very likely to have been a zoonotic transmission event in late 2019.
The next question is regarding the efficacy in children. Do we have any data yet?
Very, very little, and mainly people, as often with vaccines, are extrapolating from adult efficacy to children and licensing for children on the basis of having similar levels of neutralizing antibodies in children and adults. This is not unusual with other vaccines. It's certainly being done with new strains. So with our South African strain, Chad vaccine, we are not expected to do an efficacy trial. We're expected to show that the neutralizing antibodies induced are comparable or non-inferior, technically, to the neutralizing antibodies that you see with the original Wuhan.
The next question is very important and interesting. This has to do with the mutants that we are seeing, particularly the Delta mutant. So in order to retain the same level of efficacy, do you have to tweak your vaccine?
Yeah, we've done that. So we and many other manufacturers, it's not that difficult to make the vaccine. What takes time is manufacturing it, even for clinical trial use, and then for global supply. So that's ongoing work. As I said, a lot of work was done on the South African strain because that appeared very early this year, and that Chad South African strain vaccine or beta variant is in clinical development at the moment. So yes, you do preclinically see better neutralizing antibodies against the homologous virus strain if you make, say, a South African variant vaccine. So all other things being equal, yes, today you would like to be using the Delta variant vaccine. The problem is you can't make it in a month and have it manufactured for global supply during that time. So it takes you months and months and months to have enough vaccine made to start providing the hundreds of millions of doses that the world would like. So there's a manufacturing technology issue there, but certainly people are making the vaccines against the variants. Sure.
The next question states Covishield from Serum was approved based on data from elsewhere. Why people vaccinated with Covishield are not yet cleared to travel to the UK?
That is well, I'm not sure what you exactly mean. I think that's a UK government decision you're referring to, or are you referring to a licensure decision? There are two problems. One is access to countries, as we all know. The second is approval of the Covishield vaccine in continental Europe as opposed to the UK. It definitely was used in the UK. Doses in the UK made in Serum have been distributed in relatively small amounts in the UK over the last 9 months. So there isn't an approval problem in the UK. It has been approved. I think what the question may be referring to is the fact that the EMA have not approved the Covishield vaccine and have approved the AstraZeneca vaccine, which of course scientifically makes almost no sense because they're effectively the same product and they're manufactured to be the same product. But I understand that that will happen and is happening.
So I think the next question you've answered. The next question states that what's making the new variant more transmissible? Can we modify the vaccine in order to raise its efficacy or increase its efficacy?
Yeah, so as I said, that is happening. People are modifying the vaccine. Hopefully people will start deploying Delta strain vaccines, but it might be next year before there's vaccine available at any scale for that. The real hope with the Delta variant, even though it's much more transmissible, which is a big problem, is that it's kind of taken over in most of the world. So if that remains the situation, then if you have just a single predominant strain, that's actually easier for vaccine developers to deal with. You make a Delta variant vaccine and hope that, while you're making it, manufacturing it, and getting it out there, you've got a homologous strain virus in 99% of cases. That might be the case, because clearly that Delta is a pretty optimized viral strain that took a while to emerge, but is so much better at infecting people than other strains, that it's going to be hard for the virus to evolve something that's even better, I suspect. It's a guess, I don't know, but nothing else has really emerged to take over from the Delta. There has been micro mutations on the Delta, but it is the predominant strain globally at the moment.
The next question is, between the first and second doses, the number of weeks of gap that needs to be given has been varying, is being altered. It's been altered in India and created a lot of confusion. What are the parameters that need to be considered in order for this, the duration, the gap, to fix the gap?
Yeah, well, firstly, we understand why it's better at 3 months. You get affinity maturation of the immune response. You get memory B cells developed. When you give the second dose later, you get a greater boost of antibodies. That's not new in vaccinology. The problem is a logistic one. If you give the first dose and wait 3 months, if the first dose doesn't work well on its own, people are going to be understandably anxious that they haven't had the booster dose. We've got two really good arguments for the long interval being okay with the chimp adeno. Firstly, a very similar technology, the Ad26 human adenovirus from Janssen, is licensed and used as a single-dose vaccine. Their efficacy as a single-dose vaccine was 67%. Our efficacy as a single-dose vaccine after a single dose, I showed you, is 76%. So it looks at least as good. So during that 3 months, while you're waiting for the booster dose, you've got a vaccine of 76% efficacy against clinical disease, and much higher efficacy, of course, against severe disease. So the key consideration when you're asking, can you do a long interval, is do you have enough protection after a single dose? And our view is that with the Chad vector, the answer is yes.
The next question is, how about the vaccines from Cuba, Iran, and Turkey?
Yeah, I shouldn't comment. I don't know enough detail about them. I mean, some of these are fairly recent vaccines with not an awful lot of academic papers, peer-reviewed, published. In some cases there have been. But look, it's great that these are coming along. It's fantastic that large numbers of countries can now manufacture vaccines, often as diverse types. I understand the Cuban vaccine is a conjugate vaccine, but I think data will emerge. People are studying those.
Adrian, it's 3 o'clock. There are 3 more questions. Would you take these 3 questions?
Yes, please. Go ahead.
The next question is that now that we know that there is a lot of consensus in the RNA sequence of the coronaviruses, would it be possible for us to manufacture a vaccine that will work against any coronavirus?
I don't know. I think it's going to be difficult. We haven't got a universal flu vaccine people have been trying for decades. I think you need to remember that there are a whole variety of different coronaviruses. You know, these are beta coronaviruses we're talking about. There are two main strategies. Put in a lot of diverse spike proteins covering different strains and potential viruses, including even the mild common cold coronaviruses that we get infected with all the time. Then you might get interference between the competing antigens in the vaccine. So people are looking at that. The other generic approach is to use an internal conserved antigen like the N protein and try and get T-cell induction to the level that the T-cells will protect against that N protein, a more conserved protein. That's been tried in flu. It hasn't worked yet. I think it will work eventually, but it's not easy. So I think in the long term, yes, we should try and definitely make pan-coronavirus vaccines. We should have stockpiles of them. Even if they had 40, 50% efficacy, they'd be really useful when an outbreak happens in a localized region. But the technology is not easy.
The next question is, do regulators need fresh trials for each new variant vaccine?
They need fresh trials, but only to show safety and immunogenicity, not generally to show efficacy. That's again the same situation as in flu. We have a new flu cocktail most years. You don't need to do an efficacy trial. You need to show that you get functional antibodies in a phase 1-2 trial.
The next question is somewhat long. I have to read it out. The vehicle, the chimpanzee adenovirus, does not replicate in human cells. So the antigen spike protein gene sequences is one-time delivery, unless the second vaccine delivery may do the same. How does the memory last unless the T-cell repertoire is built up? Is there a way to improve this?
Yeah, so the adenovirus infects cells, of course. So it doesn't produce more adenoviruses, but it does express a lot of protein inside the cell, including antigen-presenting cells. And you get expression of the antigen on HLA molecules, so you get a T-cell response, and you also get secretion of the spike protein. There's a very nice paper from Bristol by Sarah's group and collaborators there, showing that you get a huge amount of expression of the spike antigen with this vaccine. So that's not hugely different to giving a protein into the arm intramuscularly. You're just producing a little factory in the muscle cell. And the durability of the response should not be less than giving a protein itself, because what you're really trying to do, of course, is to get the antigen in antigen-presenting cells in lymph nodes that will stimulate a memory B cell and T cell response. And clearly that happens. So I don't see any intrinsic reason why using a DNA vaccine or an RNA vaccine or a viral vector that have to produce the protein in the body should give less durable memory than giving the antigen itself or giving an inactivated virus. And I think the data that we have now show that that is indeed the case. There is good durability with those sort of encoded antigens as vaccines.
Well, there are more questions coming up, but I think we should stop now. There are many comments that actually thank you for a clear presentation of this entire area, especially the manufacturing of the Chadocs. On my own behalf, on behalf of the Fellowship and Associateship of the Indian Academy of Sciences, I thank you for your consent to be with us this morning and give this wonderful, clear presentation. We hope that one of these days we can bring you to India and have you give a lecture in our academy, and that would be a fantastic to interact with you and hear you live as opposed to on the screen. Thank you very much, Adrian, and really grateful for this talk.
Wonderful. Thank you for those kind remarks and for fantastic questions and your interest. Bye-bye.